So today we'll start off with a bit of a literature review from Scott on the differences between species in connectivity and cortical column structure.
So after Focus Week, during Focus Week, I was thinking more about uh, the fact that other animals have columns, but they're not nearly as smart as us. There's mice, there's rats, monkeys, and I was starting to think about, how much can a column do? Can human columns do a lot more than rat and mice columns? And how much should we expect a column to do? I know we tend to kind of take a maximalist sort of position. I think I don't know if anybody would disagree with that, but take more of a maximalist position on what columns are capable of. And so I just started thinking about, what are columns capable of in a mouse or rat or a mouse or rat. And I've always been sort of drawn to this idea that our amount of neocortex has really exploded in recent recent years, recent millions of years in terms of evolutionary time. So the neocortical explosion. And so this is sort of a more columns view. The reason why we're smarter is just because we've got more columns. That's pretty straightforward. There's no real need to go into that. Um, but I was also interested in just, what are the actual differences between our columns and our relatives, our cousins. And um, so this is just a quick foray into the literature because I just hadn't spent a lot of time with this kind of stuff. And so
Is this a literature on the anatomy of columns, or
a
literature
bit,
about,
yeah.
or is it more
And
about people speculating why humans are different than other mammals?
this is mostly going to be just talking about some anatomy, like
So, yeah.
difference
So
in
that
pyramidal cells and things like that.
Okay. So before you go there, just to complete this picture, there's another hypothesis about that it's in between these two, and it has a lot to do with you could have similar columns. We do have more columns, but it's also how those columns are applied. It can make a big difference. So one thing that a lot of scientists have proposed, or not a lot, but some have, is that there's a role of language. And language requires that we have the ability to generate language and to be able to understand it. And when I say generate it, like physically generate it. So I read one paper once. It was a hypothesis about the difference between monkeys and humans, and there's this projection from part of the cortex down to the sort of the voice box area, and it's kind of missing or very minimal in a monkey.
Mhm.
And so they wouldn't have the same ability to articulate sounds that we do.
Right.
So that could be a big basis of like, hey, monkeys might have pretty sophisticated thoughts, but they have no way of talking to each other, or at least not in any sophisticated way. And so it kind of limits what they can learn. And then other similar hypothesis about the opposable thumb. People say, well, you have to have a really manipulable hands to do things. So there's an in between more columns and better columns. It's like better physical apparatus attached to the cortex. That's
Right.
all right. So, but
And
doesn't, yeah.
Well, since you mentioned that, I would just say one fun fact that I found, which is that a lot of people claim that we've never seen an ape ask a question. They understand questions. Like if you ask an ape a question, oftentimes we'll actually use the sign language to repeat the question and then answer it. But a lot of people say, it never asks, what is that? Who is that? Things of that nature. Questions that it can't understand. And this is just kind of a weird fact. Um, like just a strange. All
Is
right,
that
but
is it,
I'll just leave that as it is
okay? Leave
because
that as
because that's just a
yeah.
weird thing that I came across.
it's
Um,
interesting.
I
yeah,
assume you meant they're talking about like apes who've learned sign language. Is that right?
yes, apes will learn sign language, and like I said, they understand questions because they can answer them, but
Uh,
they're not out there asking. At least in
there
any
is a
way that sometimes they might point at a banana and that kind of suggests they're asking, "Can I have that?" But it's nothing like the kinds of questions that they're asked and able to successfully answer.
yeah.
But it's like, I mean, animals definitely have like curiosity, and especially young animals do engage in play, which doesn't have like any particular purpose besides learning about things and learning how to do things. So it's not like they don't have hypotheses and test them or want to learn things or are curious about stuff, but maybe they aren't able to use language to express those, or
Yeah.
maybe they're not young enough to be interested in asking questions anymore at the point that they have learned sign language.
Yeah.
Yeah.
I don't really know what to make of it.
There's another thing too, like some animals like whales have huge brains and they have huge number of columns. But they're all sort of, they're more like primary type of regions, you know what I'm saying? It's like, so like we've often talked, like it seems like rodents, like mice have a single visual region. Like they have one V1, they're on V2 and V4, that kind of stuff. And so they can still see, but then it would limit them to represent compositional structure of any kind of sophistication, right? They might see everything just as morphology. So there's a whole bunch of nuances here between like, it's not, I mean, the number of columns, how they're arranged is a big difference, is it hierarchical and so on, and then what are they connected to physically is a big difference. So it could be as you know, they've been saying the animals are very, or you're saying they're very curious. Would they ask a question if they had some way of doing that?
Yeah, I was gonna
Yeah.
say, besides the two, like more columns and better columns, it seems like there's a third answer, which is better connectivity between columns, like whether
Right.
it's stacking them more hierarchically, instead of just having a very flat number of columns, or different kind of more context signals to L1 or whatever, or better connectivity from planning regions to lower sensory regions. So
You
yeah.
know, and if a human is raised without any language at all, they have a tremendous difficulty doing it later. And so, you know, even if an adult ape doesn't seem to understand language, doesn't mean it couldn't understand it if it had raised in an environment where it could hear and talk, you know. So there's a lot of subtlety to this, but I think we're getting off track. I think it's useful to look at the anatomy differences between species.
Yeah. So I just want to say, as it's been hinted at, this is like a vast area of research and literature. This is just like me spending a little bit of time looking at a couple things in particular. So first thing I was looking at was just pyramidal cell morphology and and this idea of dendritic scaling. So this is basically what Vivian was suggesting as a third option. As the brain grows, there needs to be more ways that they communicate with each other. So one might expect that pyramidal cells, for example, just grow uniformly, or their dendritic number of dendritic compartments grow, synapses and things like that. And this is pretty interesting and I think well reproduced finding, which is that if I just look at, say, marmoset here, which is um, you know, a lower primate, quote unquote lower, and then macaque and human. For humans, if I take V1 and look at, say, number of spines, the difference between V1 and like an associative area and PFC, the number of spines in a layer 3 pyramidal cell changes substantially,
Well, this is
very
just because
substantially.
this is the spine density,
Yes.
which is not the same as number of spines. You could imply the number of spines, but because the branching of the human might be bigger too. So if I were reading this, I'm not sure. This says per 10 microns. That would, I assume, mean 10 microns of a dendritic branch. And so, like, how many synapses you have per micron, you know, per 10 microns. So it's more synapses. Yeah. They're closer spaced on the dendrite, but also the dendrites could be much larger. Well, I think they're generally up there. Next thing up, I guess, basal dendritic area, right?
Yeah.
So you have to multiply those two.
They tend to not grow too much in V1. So I'll come back to the previous slide. So you have to kind of synthesize different bits of literature because some will have humans versus monkey, and some will have monkeys versus mouse and that kind of thing. And so this is a mouse versus monkey paper. If I look at mouse V1 and monkey V1, scaling is about the same. Roughly the size is about the same. They say V1 is relatively conserved in this respect. Yeah, OJ did her PhD on morphology. She has a lot, I'm sure, to say about this kind of thing. But if I look at the mouse V1 versus mouse frontal cortical cells, they're also about the same, roughly. So the amount of specialization in the higher cortical areas is pretty mild in quote unquote lower animals versus, say, um, monkey or human. The amount of it's just far more elaborate as you move up the cortical hierarchy, what these cells are like. So it's not really a simple answer. Like our dendritic scaling isn't just increasing with the size of the brain. It's also dependent upon the area and more specifically, sort of where you are in that cortical hierarchy. Does that make sense?
Yeah, I think it's worth pointing out though, if we think about our models of the neuron, so you have dendritic integration along the basal dendrites and the apical dendrites, the spikes and there's almost no, well, I don't know, I would say no evidence. I'm not aware of much evidence to suggest that the mechanisms of these cells are different. In fact, most people assume that a mouse or a monkey pyramidal cell works on the same principles as a human pyramidal cell. So we're really just talking about numbers here. It's important to point that out. This complexity doesn't, it allows the cell to recognize more patterns, but it doesn't do it in a different way. That's my working assumption.
Yeah, I was also interpreting the more elaborate dendritic arbors in PFC as just reflecting the amount of things that come and go from it as well. You just need more space if you connect into more neurons.
Yeah, so
that kind of,
in our case, remember, it's not integrating lots of all these things. It's the number of patterns it can learn. Remember, because on all these, a 20 micron segment of dendrite is what you need to recognize a pattern. And so you have to, 20 to 40, 20 to 40.
Right.
40
I don't think they're incompatible though. Like you can learn patterns that come from more sources.
Right, basically. Right. But my point is, it's really important to think of it as you're increasing the number of patterns that the cell can associate with. It's not necessarily increasing the complexity of the patterns. It's just saying, oh, if I could, a simple cell might be able to recognize, 500 different unique SDRs. Well, a more complex one might be able to recognize 5,000, that kind of thing. But they're processing it in the same way.
Right.
That's the assumption.
So I'm just going to go back to the V1 or, this previous slide, which is like when it says occipital, you can just think V1 here and frontal, more or less PFC and in a monkey or whatever kind of frontal area marmoset has. I don't know if they call it PFC. But if I look at, say, basal dendritic field in human, it does increase a bit. So I'm looking at this number here, the basically amount of basal dendrite. There is an increase, but it's, I don't want to say it's modest. It is what it is. All these animals tend to increase their basal fields slightly, except for marmoset, which has this kind of strange pattern. But if I look at spine density in humans, that's really scaling up pretty substantially as you move up the cortical hierarchy. So I say the amount of basal dendrite in prefrontal cortex is roughly the same in macaque as it is with humans, but the spine density is much larger in humans compared with macaques. So I think that's just a part of this picture that I found while just looking into morphological changes as a function of species, which was just kind of an interesting thing to keep in mind.
Could you go back to the figure in the previous? Yeah, this one. No, the one with the morphology. Are the different columns here just different examples, or are they like specific?
Yeah.
Okay, cool.
They're just different examples.
You have to also, you have to be really careful here too, because there's lots of pyramidal cells. So here they're talking about layer 3 pyramidal cells, right? That's just only one. I mean, we got layer 6 and layer 5 and layer 2, and so. I mean, we can walk away with some general ideas here, but we have to be careful reading too much into it.
Yeah. Let's see what else.
Can you show the numbers? The next slide here. There was something interesting on there. This slide, yeah. There's something I hadn't realized here. How many synapses there are on a pyramidal cell? And so people say, oh, there's thousands of them. And a typical number I would hear, over the years, I don't know what year this paper was, 2001, but over the years, someone would say, well, somewhere between like 3 and 10,000 synapses per pyramidal cell. And then I read this paper which very carefully counted the number of synapses on pyramidal cells in the hippocampus, and it was over 30,000. And so I said, oh, I always thought there was like, okay, in the cortex you have like 3 to 10, and then the hippocampus you got 30,000, and we have a theory about that. But here was something I didn't realize here. They're saying here the temporal lobe and the frontal lobe are, well, they're showing over 12,000 for a human. That's surprising to me. I didn't know that. I didn't know there's that big a difference between V1 and and the temporal lobe. I didn't realize that the V1 these cells would be that small, that few synapses. So the thing I walk away from this is it's not like you just have this big bump when you get to the hippocampus. Is that when you get away from, perhaps you could read into this, when you get away from the primary sensory regions like V1, then go up the hierarchy, it's getting many more synapses as you go. So the memory capacity of higher regions could be much, much higher than lower regions. They're actually getting smaller, the higher regions. The regions V1 is huge. And we know why, because it's got many models. But this tells me that the columns themselves, if a single cell can have 14,000 synapses, that tells me that the columns in these upper regions are they have much more memory capacity than I assumed. And so in the past, I've done a number of calculations, like how many patterns could a cell recognize. It sort of puts a limit on what a cortical column could do. And I assumed lower numbers. I didn't assume ever over 10,000. In fact, I think I usually assume something around 5,000. So that tells me that these columns have, upper regions could have much more capacity per column. Same function, doing the same thing, but just a lot more memory.
Yeah, it makes kind of sense, maybe, in the sense that you can only have a certain amount of direct sensory, a certain amount of variety in the sensory input you get. So you don't need that much representational capacity to represent all those patterns. But then once you go up the hierarchy and you can actually learn compositional models, there's basically an infinite variety of ways to combine different objects with each other. So you need a lot more capacity to represent all these different compositional models that are relevant to you.
Yeah.
So maybe that's why
it's
necessary.
I have a maybe similar, but I think it's a different hypothesis as might also consider. When you look at the hippocampus, and I've shown that picture of the tank paper several times, and they show what a grid cell looks like in the entorhinal cortex. And it looks like it almost looks like the hippocampus is designed where there's like one column per layer. It's like there's no more regions. It's like, it's like you in V1, you have a region with many thousands of columns. And then when you get up high enough, like in the hippocampus area, it's like almost everything's being done by one column. And so the other hypothesis would be the following: is like the reason you have to have lots of the whole point of having lots of columns and them voting is you can do fast inference, right? You can do this flash inference. You have many models that are looking at all these things at once. But once you know what the object is, once you said, oh, that's a dog, you've done this, you need many columns to do that quickly. Otherwise, you have to move your eyes a lot, right? So we have many columns. They're going to quickly reach a consensus of what the object is. But once you know what the object is, you don't need a lot of columns anymore. It's like voting becomes not so important. Maybe you're voting now between modalities. But the whole idea that these big V1 and V2 and these big S1 and S2, which are huge, could be just because the brain wants to be able to quickly know what it's looking at. And once it knows what it's looking at or sensing, it doesn't need this broad array anymore. It doesn't need to do a lot of voting because it's already done it. So that's why you might see these. You would need once I know what the object is, I don't need to have a lot of columns voting on anymore, and I need a lot of capacity in the columns that are manipulating the known object. I don't know if that makes sense. But I view that as these very large primary, secondary regions are really just big, so we can do fast inference.
Yeah.
And then it goes away. Anyway, I like this result. This is an interesting point. I'm going to start thinking like, oh, upper columns could have 15,000 synapses per cell.
So this ties into one other, I have just a few other facts here. One, the large like basically visual acuity scales with the size of E1 and the number of columns there. So I think this fits in nicely with it. So I'm kind of thinking of primary sensory areas as benefiting more from the more column. If we're doing more columns versus better columns as sort of a spectrum, I'm thinking of primary sensory areas as fitting the sort of more column further down that spectrum, and higher cortical areas sort of in the better column spectrum.
Or I would say
Very broadly.
I wouldn't say better, say higher capacity.
Higher capacity, more elaborate, but also like more elaborate in terms of its inhibitory networks also, which I think is next slide. So, you know, I don't know what to think about the inhibitory picture. It gets a bit more complicated in humans than it is relative to monkeys and relative to mice.
Right. That's an area where you do see a lot of much more variation, I'd say, than with the excitatory cells.
Yeah. Sorry, go ahead.
Does the dendritic scaling correlate to more neurons, or like is it necessarily more neurons, or is it just more connections between the same number of neurons?
No, not necessarily. Mice tend to have, you know, their cortex is like 1 millimeter thick versus ours is about 2 millimeters thick. And I think there's been some back and forth on this. Jeff, you might know better than me on this, but in terms of number of neurons in a column, I think there's been some back and forth in the field about that. I think it's relatively conserved. It's just more densely packed in the mouse.
Right, they're more densely packed. And so how many cells you can fit in determines based a whole bunch of reasons. How big are the cell bodies? How big in diameter are the dendrites? Are they myelinated or not myelinated? And how density is the branching? So all these things come into play. My recollection of what you just said, Sky, is that they're relatively preserved. You know, the number, it's not like dramatic number of neurons change in the column. It's more like, oh, they're smaller. You know, in a mouse, if you need bigger cell bodies if you have longer dendrites and longer axons, because the cell body supports it metabolically. So if everything is small and compact, then you can have smaller cell bodies. And then you can have narrower dendrites because they don't conduct as well, but they don't have to go as far. So all these things compact that you can get a lot more stuff stuffed into a mouse column than you can into a human column. We have big, fluffy, you know, flowery neurons. You have to kind of visualize it like that because they have all these connections, and the mouse is like, wait, break it all down.
That is a true statement, yes.
What's that?
Yeah, and that is a true statement from like when I was studying neuromorphology.
I should maybe you're the expert here. I should
yeah. Well, I mean, I study mostly like interneurons, and like the pyramidal cells were kind of secondary. But in general, like there was like one part of my project which was like, you know, trying to generate or like predict what the human like pyramidal cells would look like but after training like a model on the mouse. And long story short is it could not really do that because there's too much difference in the morphology, but in particular size. So human ones are like larger in terms of, especially in terms of the length of their apical dendrites. I mean, again, we have a bigger like cortex, you know, 2.5. We need to stretch longer than the mouse, and also the apical tuft. So like the stuff at the end of the apical dendrites are more extensive branching. And we also have a bigger cell soma. So like when I train a model on the mouse, it couldn't generate all those like characteristics because, you know, I mean, the striking one was kind of like size, but size in like many different things like soma, the apodendrite length, and that affects the branching points.
Right. How thick are the dendrites? And how are they myelinated? Because myelin makes you faster. All these things come into play. The general thing I've walked away with of looking at this for decades is that, you know, it's a reasonably good assumption to say that the functional description of columns is going to be very close between all these different mammal species, but there's a huge variety in the capacity and the number and how they're arranged and all that kind of stuff. And this is fundamental to our work, right? We've been under this assumption from the beginning that a mini column is a mini column, and you know, if that's not, I mean, a column's a column, and if that's not true, then you know, but it is true. So, but if it wasn't true, it'd be bad.
Yeah.
Make our life hard.
So this first bullet point here actually touches back on something you were talking about, Jeff, with language areas. Our columns are a bit wider. Our mini columns are a bit wider.
This is mini columns now. Are they really talking about mini columns?
They are talking about mini columns.
Okay.
And I think they're mostly making the argument that they're wider due to increased neural pill.
Right.
I'm not sure.
Right.
So for the unfamiliar, neural pill here, we're talking about white matter tracks and things like that. So
neural pill, I think it's mostly just referring to axons and dendrites. I don't think it includes cell body. Is that,
yeah.
Yeah, which is consistent again with more connections.
Right. And the biggest difference here is if we look at language areas between, say, humans and gorillas, our mini columns are actually quite a bit wider, which is an interesting.
Do gorillas have language areas?
No. Well, I mean, they have the same brain area, but
Okay.
the language area is for us, but for them, I don't know what they're actually used for.
Interesting. Well, yeah, a lot of this could be also determined by usage. I mean, the brain is extremely plastic, right? Yeah. And so, you know, you can, when you train on something, that part of the brain gets bigger. That part of your neocortex gets bigger. The neurons send out more branches. And this is a real physical thing, and it's not minor. So, you know, one that's 30% wider, it could be just like, you know, it's because we talk. It wasn't like, it may not even be, you know, genetically determined. It's just because, you know, we talk a lot. Yeah. And, you know, they showed that with we've talked about this study where they showed that the British taxi cab drivers, you know, had to learn the map of the city, and that part of the hippocampus became bigger because they just trained it, you know. Yeah. So, you know, what's genetic and what's learned is hard to say.
I didn't include the citation here, but I think it was from the Gilman paper. But that's pretty much all I have. I mean, we touched on the inhibitory complexity, and I know double bouquet cells have come up here before, and those are inhibitory cells that you know, sort of span the mini column, I guess. But if you're interested in any of these papers, they're up on the Zotero